Greetings to you all. My name is Nancy Jones, and I am the Vice President of Clinical Development at Neuren Pharmaceuticals. It's a great pleasure to join you remotely and give you this update. First of all, let me introduce you to Neuren. Neuren is a biopharmaceutical company specializing in the development of drugs related to neurodevelopmental disorders. We are a small team, and we're all dedicated to finding new treatments for patients who are affected by a rare genetic disease and have great medical needs, but to date have no specific treatment. The table gives an overview of our clinical program. We have two new drugs in development. NNZ-2591 is in development for 4 genetic disorders in phase 2 clinical trials. The first molecule is called Trofinetide and is our most advanced program. We have completed a phase 2 study for Fragile X syndrome. As you can see, we are evaluating Trofinetide as a potential treatment for Rett syndrome. And a phase 3 clinical trial has been completed. A phase 3 clinical trial is a large study that's designed to provide the evidence for regulatory approval. It was our partner for North America, Acadia Pharmaceuticals, that conducted this phase 3 clinical study and submitted a new drug application, or an NDA, which is the application for approval of a new drug in the United States, and that was submitted to the Food and Drug Administration, the FDA. So what is Trofinetide and why might it be useful for the treatment of Rett syndrome? So before talking about Trofinetide specifically, it is useful to talk a bit about insulin-like growth factor, or IGF-1, and to understand how IGF-1 works in the brain normally. So on the left is a picture of the cells in the brain. The orange cells are the neurons, which are the main cells that you have for functioning of the brain. And there are other cells that support the neurons, notably the microglia that are shown here in brown and the astrocytes that are shown here in green. The microglia help with the growth and also the maintenance of the structures of the neurons and also the function of the neurons. And this includes the dendritic spine. So those are those brain things that branch out from the neuron and they receive the messages from other cells. So naturally in the brain, all of these cells produce IGF-1. And IGF-1 is a growth factor. It's produced throughout the body. And in the brain, it's responsible for the growth and the regulation of neurons. And it's also responsible for the communication or the connection between those neurons, which are called the synapses. So here we see the IGF-1 molecule. And when it does its job naturally, it separates into smaller parts, or what are called metabolites. And one of these small parts is a GPE. And GPE is the terminal tripeptide of IGF-1. And what Trofinetide is a synthetic analog of GPE, and it basically replicates the function of GPE. So what's an analog? An analog is something that has almost exactly the same structure, chemical structure as the naturally occurring molecule, but has one small change. And here you can see at the bottom the chemical structure of GPE on the left and Trofinetide. And they're exactly the same, except there's addition of a methyl group, as you'll see on the right, for Trofinetide. And one question people may ask is, well, why wouldn't you then just use GPE as a drug, as a medication? And that's a good question. So essentially, what are the benefits of this change? So what this change does is that gives Trofinetide better characteristics to be a drug or a medication. And in this case, for example, it stays longer in the body. So if you see on that the graph on the bottom, this is an animal model in a rat, and GPE was found to last for 5 minutes. But Trofinetide lasted for 74 minutes, which is more advantageous because it would mean that you would be required to take the treatment fewer times per day because it lasts longer in the body. And that's just one example of sort of the properties that can be improved to make it better to use as a medication. Another important question, obviously, is the relevance of Trofinetide for Rett syndrome. And we do know that it has been observed that there are insufficient formation of the new synapses by neuron, those connections, and then excessive pruning of the synapses by those overactive microglia, those supporting cells. So Trofinetide is thought to improve synaptic function and restore synaptic structure. and it inhibits overactivation of inflammatory microglia and astrocytes, so supporting cells, and then also to increase the amount of IGF-1 in the brain. In preclinical studies, we have also specific evidence for Rett syndrome. So I'm showing here the results from a mouse model. And in that mouse model, we found that GPE treatment improved the effects of Rett syndrome. So specifically in this study, it increased lifespan, brain weight, and also the density of those dendritic spines. And then on the right-hand side, you see in a cell-based study, so this is looking at the effect on cells, we found that GPE and IGF-1 can correct neuronal deficits caused by MECP2 mutant astrocytes. So supported by this information, 2 phase 2 studies were conducted. And phase 2 studies are studies to assess the safety and provide the indications of efficacy. So on the left here is the findings from study Rett 001. Rett 001 was a double-blind, placebo-controlled study with 56 participants, 60 to 45 years old. And participants were randomly assigned to receive 1 of 2 doses of trofinetide or placebo for 28 days. Trofinetide was well tolerated, and there were encouraging trends for efficacy. The Rett 02 study was a double-blind, placebo-controlled study with 82 participants between the ages 5 and 15 years old. Trofinetide was also well tolerated in the study, and for the highest dose, we observed improvement in 2 assessments. That was in a questionnaire for the caregiver, which assessed key Rett symptoms, and also in the clinical global impression of improvement, which is a clinician measure of overall improvement. Daffodil is another study, a phase 2 study, which is an open label study looking at young children aged 2 to 5 years old. The goal for this study is to evaluate safety and pharmacokinetics. And pharmacokinetics means learning how the drug is used and absorbed by the body. And this study is ongoing, but no longer enrolled. The phase 3 program consists of 3 studies. Lavender, and that is the controlled double-blind study. Lilac, which is the open-label treatment study. And then Lilac-2 is a study for participants who complete the Lilac study, but can continue on the drug until the new drug application is reviewed. So Lavender was randomized, double-blind, placebo-controlled study. The duration of treatment was 12 weeks. The study included 187 girls and women aged 5 to 20 years old. The co-primary efficacy assessments were the RSBQ and the CGI-I. Secondary efficacy assessment was the Communication and Symbolic Behavior Skills Developmental Profile, specifically the Social Composite Score. That's the CSBS. The CSBS is an assessment of early communication behaviors. This includes emotion, engage, gestures, communication of needs and requests. And the study was conducted in the United States. So a little bit more about the co-primary efficacy measures. So the Rett Syndrome Behavior Questionnaire is a validated 45-item rating scale. It's completed by the caregiver. And the evaluation covers 8 areas specific to Rett. It's scored from 0 to 2, and higher scores indicate more impairment. And this measure has been correlated with functioning and quality of life in Rett. The Clinical Global Impression Scale of Improvement, or CGI-I, is a global assessment of a change completed by the clinician. The scores range from 1 to 7, with scores of 1 to 3 indicating improvement, 4 indicating no change, and 5 to 6 are scores that indicate worsening. The overall findings for the Lavender study are presented here. There were statistically significant separation from placebo on the 2 co-primary endpoints, and there was also statistically significant separation from placebo on the key secondary endpoint. I'll talk now in more detail about the Lavender study. Here we see the summary of the participant characteristics. So the treatment groups, the different treatment groups, placebo and trofinetide, were similar for age and also the baseline severity on the CGI-I, which is a global impression of severity. And just over half the girls were between 5 to 10 years old. Just over a quarter were between 11 to 15 years old, and about 20% were between 16 and 20 years old. And based on the CGI-S, the majority of participants were moderately or markedly ill, and very few were considered extremely ill. Here are the results for the RSBQ and the CGI-I in detail. We observed a statistically significant improvement in RSBQ compared to placebo, and we observed a statistically significant improvement of CGI-I in the trofinetide group compared to placebo. For the secondary endpoint on CSBS, there was a statistically significant separation from placebo. So what types of symptoms changed? This can be seen on the RSBQ, which has subdomains in addition to the total score. And here what you're seeing on the graph is a measure of the magnitude of effect or the difference in magnitude between the placebo group and the trofinetide group in each of these domains. The direction on the left indicates a change in favor of trofinetide. And as you can see, all the subscales were directionally in favor of trofinetide. And another important point is to note that the effect was not actually due just to a single subscale. Here we see the findings for safety. The most common adverse events were diarrhea, observed in 81% in the trofinetide group versus 19% in the placebo group. In the trofinetide arm, of those that had the diarrhea, 97% were characterized as having a case that was mild to moderate. The next most common adverse event was vomiting, 27% in the trofinetide group and 10% in the placebo group. And in the trofinetide arm, 96% were characterized as having cases that were mild to moderate. Serious adverse events were observed in 3% of the study participants in both the trofinetide and the placebo groups. So what is the status of the new drug application? Acadia submitted the new drug application in July of 2022 for the treatment of Rett syndrome in patients 2 years of age and older. The FDA accepted the NDA for a priority review, so the decision date set for the 12th of March in 2023. The decision will be based on review of the pivotal efficacy, which is the positive phase 3 Lavender study, supportive efficacy, which is from the phase 2 trial in Rett syndrome, and then also the safety data from the completed and ongoing studies. As of the date of the recording of the presentation, the decision date had not yet been reached, and an update on the outcome of the review on March 12th would be announced on our website. And what is the current situation for the European market? Neuren retains ownership of trofinetide outside North America, including Europe, and Neuren is currently in discussions with potential commercial partners for Europe. We intend to apply for a marketing authorization for Rett syndrome for the European Medicines Agency, the EMA, using the results from the US program. We have received scientific advice from several national regulatory agencies, which indicated the steps to follow to increase the probability of a positive approval. So I'd like to end by acknowledging the efforts of the clinicians and the study teams that participated in the studies I presented here. And of course, very, very special thanks to the girls, the women, and their families who participated and are participating in the studies. I thank you for your attention, and I wish you to have enjoy the rest of the conference.